Welcome back, my dear students. This is Mabo Sultanaki, Senior Lecturer of Manston College, Department of Biology. Today, I will discuss Chapter 1. From this chapter, I will discuss some knowledge-based questions. These knowledge-based questions are from three topics. One is replication, transcription, and translation. Besides this, I will also discuss what is genetic code, characteristics of genetic codon, all these I will discuss. So from today's lecture, you will be able to write the definitions of these knowledge-based questions. Along with this, you will be able to explain what are the characteristics of genetic codon and the function of genetic codons. Okay. Now, first definition is gene. What is gene? The region of DNA which carries or which controls different characteristics or particular characteristics of an organism is called gene. Then cistron. Actually, the functional unit of DNA is called cistron. You can write this definition in this way also. The part of DNA which controls the functions of cellular substance is called cistron. Next, record. What is record? The combinational unit of cistron is called record. Then muton. The mutational part of cistron is called muton. Then replicant. What is replicant? The part of DNA which controls DNA replication is called replicon. Now, promoter gene. What is promoter gene? The specific base sequence in the DNA at which DNA polymerase binds is called promoter gene. Exon. The newly formed pre-mRNA has some regions that contain genetic masses, which are called the exons. Now, introns. In the newly formed pre-mRNA has some regions which do not contain any kind of masses or genetic masses, which are called introns. Operon. A structural gene, an operator gene, and a promoter take part in protein synthesis, which are all together known as operon. Now, genetic code. The group of nucleotides or nitrogen base for bases forming the signal for an amino acid is called genetic code. You know what is nucleotide? A ribose sugar, nitrogen base, and inorganic phosphate combining or collectively form a unit which is called nucleotide. So the group of nucleotide or nitrogen bases which that is formed for the signal of an amino acid, that is called genetic code. You know there are 20 types of amino acid. Codon. The genetic code consists of 64 triplets of nucleotides. These triplets are called codon. Now, what are the codons? You know, there are two types of nitrogen bases, purine and pyrimidine. Purine, adenine and guanine is known as purine. Cytosine, thymine and uracil, all these are known as pyrimidines. So, if AUG or UAG, these triplets form any particular unit, that is called codon. Okay? Now, start codon. What is start codon? The codon which starts the translation process is called start codon. This start codon is indicated by AUG. This AUG codes for methionine. Now, stop codon. What is stop codon? The codon which provides the signal for the termination of polypeptide chain is called stop codon. There are three stop codons: UAA, UAG and UGA. Now, the characteristics of genetic code. There are several characteristics of genetic code. First of all, the genetic code is triplet code where three adjacent bases of mRNA form a codon that specify one amino acid. For example, if the mRNA is of UAA, C, U, G, C, then these three will form a triplet codon. Okay, the adjacent three. Okay. Next, genetic code always non-overlapping. Adjacent codons do not overlap each other. Number 3, there is no punctuation in the genetic code. That is, genetic code is commaless. My dear students, you can see that there is no comma or no punctuation in this among these genetic codes. Then genetic code is universal. It means that a particular genetic code or particular codon indicates the same amino acid for a particular protein synthesis. The genetic code lacks specificity and one amino acid often has more than one code triplex. This phenomena is known as redundancy or degeneracy. Next, each code codes for only one amino acid, none for more than one. 3 of the 64 codons names UAA, UAG, UGA do not specify any amino acid, but it provides signal for the end of the message. These are known as nonsense or stop codon. The codon AUG is called the initiation or start codon as it begins the synthesis of polypeptide. The genetic codes have definite towards the 5' to 3' polarity. So my dear students, these all are the characteristics of genetic code. So from my today's lecture, you will be able to write different knowledge-based questions along with the characteristics of genetic codon. These are all for today. In my next class, I will be back with a new lesson. Till then, stay safe and healthy.